It's a big challenge for me to give this talk for young scientists. Although I have declared in the title the biophysical aspects of the theory of molecular solitons, I will give you also some information about application of this approach to in a more general sense. First of all, my sincere thanks to Sergei. F5 on my computer. This is F5. Yes, yes, yes. Who did a great job and very important job in this very difficult time for Ukraine and especially for young scientists. I will tell you about some investigation done in collaboration with my colleagues, Alexander Yeremko from my institute and from this international team. The next slide, please. Let me remind you that in a few days, on 26th of December, we will celebrate 110 years since the birth of Alexander Davydov, a great scientist, physicist, theoretician, who worked in various areas of theoretical physics. And next year, we will celebrate 50 years of Davydov's Soliton. Since we are from different subjects, let me remind you what is a soliton. A soliton is a solution of nonlinear equation or a system of equations in the form of a solitary, localized, dissipatively wave. And the peculiarity of solitons is that when they interact with each other, they interact almost like particles. They do not change their form and velocity during the collisions. And the particular type of soliton, which is called Davydov or sometimes Davydov's soliton, is based on the account of the polaron effect. The next slide, please. We know that solitons exist in various systems, in atmosphere, like you can see the morning glory clouds. In Wikipedia, it says that we can watch this in northern hemisphere, in the northern west hemisphere, but I myself watched such clouds in Kiev over the Dnieper River in the late afternoon, on the also on the western part of the sky. The lower photo shows the soliton wave, solitary wave in the Black Sea. It can be tsunami, like you can see on the right-hand side, and it can be in the form as it has been for the first time observed. This is well-known picture of the reconstruction of the solitary wave in Edinburgh Canal. The next slide, please. Davydov soliton. There was a crisis in biophysics that very low basic unit of energy in biological systems, which is the energy of the ATP hydrolysis, is released in one part of the cell and is utilized in a remote part of the cell. And this occurs at physiological temperatures, which are high temperatures, which we know, as we know. And this amount of energy is very, very low. So the crisis of bioenergetics. And Davydov has suggested the following idea, that this process of the energy transfer in the cell is essentially facilitated by the participation of polypeptide macromolecules. Polypeptide macromolecules possess periodical crystal-like structure. They are stabilized by the hydrogen bonds. They are soft, that is why they can form the globular structures. And this means, from the point of view of the solid state physics, that electron lattice interaction in these systems is very essential. So the simplest model Davydov and Kisluha suggested was to consider an isolated polypeptide chain, like shown on this figure, where the black dots are peptide groups, connected by the hydrogen bonds with elasticity parameter kappa, and electron lattice interaction strength is characterized by the parameter sigma, and resonant electron interaction or exciton interaction is characterized by the parameter J. So, amide-1 vibration, which is the energy of the hydrolysis, is mainly stored in the excitation of amide-1 vibration, which is essentially the vibration of the double carbon-oxygen bond. This vibration interacts with lattice, deforms the lattice, creates a polaron effect, and together they form a self-trapped bound state called later Davydov soliton. And in this bound state, the deformation and amide-1 vibration can propagate along the polypeptide chain with constant velocity without energy dissipation in the continuum approximation. Later on, this approach has been generalized for the processes of electron transport, and electron transport occurs in biological systems, in respiration and photosynthesis, so-called the redox processes. The next slide, please. Such system is characterized by the Fröhlich Hamiltonian, as you can see, which consists of three parts, three terms. The Hamiltonian of the electron lattice, of the electron subsystem, the Hamiltonian of phonons, and their interaction. From this Hamiltonian, we can derive the system of equations for the excitation probability. I will say a quasi-particle because it can be also electron, as I told you already. Another part is the Hamiltonian for phonons and their interaction. So we can derive the system of equations for electron subsystem, for lattice subsystem, and in the continuum and adiabatic approximation, this system can be reduced to the nonlinear Schrödinger equation, the known solution of which is the soliton. There is an essential parameter in this equation, the nonlinearity parameter, G. This parameter is proportional to the square of the electron lattice interaction, and it is proportional to the elasticity of the chain and resonant interaction energy. Depending, this parameter G defines the width and amplitude of the soliton, because the wave function of electron psi, which is in this equation, in nonlinear Schrödinger equation, is normalized by unity, as quantum mechanics teaches us. So depending on the value of parameter G, the amplitude of this soliton can be higher or lower. The solid line, blue line, shows the envelope of the soliton at the initial time moment, and the dotted line sometime later. So you will see that the soliton propagated for at certain distance along the chain. And the red curve corresponds to the value of nonlinearity parameter G equal 1. The next slide, please. We know we can calculate Soliton wave function is well known. This is the Bell cosine, hyperbolic cosine function. But you remember that the soliton is not only the electron or emit1 excitation, it is also the chain deformation, which is proportional to the psi squared. We know that we can calculate the total soliton energy, taking into account the kinetic energy of its propagation. What is important is that soliton has effective mass which is higher than the mass of an electron or emit1 excitation because of the dressing by phonons, because of this polaron effect. And the soliton energy is lower than the energy of the band bottom. That is why this is the stable solution, plus there is no dissipation of energy. And that explains the possibility of this energy transfer or electron transfer on macroscopic distances. The next slide, please. But in the respiration or in photosynthesis, in the redox processes, there is not one electron released, but 4 electrons, 2 plus 2 electrons. 2 electrons, now I speak about electrons, not about emit1 excitation. 2 electrons, each electron has a spin. In the absence of the magnetic field, it is energetically advantageous for 2 electrons to form a bound singlet state. They form a single deformation in the chain, and both electrons occupy the bound energy state, energy level in this potential well. As a result of this binding, the amplitude of bisoliton is 2 times higher than the energy of a soliton isolated, and respectively its width is twice narrower. And its energy is much lower, 8 times lower than the energy of the soliton. If there is relatively strong magnetic field, which can reorient spins so that they become parallel, then this singlet bisoliton state is forbidden. And in the result, the electrons form triplet bound state. It is still a bound state. Its energy is lower than the energy of the 2 free electrons. And on this curve, you can see a red solid line. This is the bisoliton. The blue solid line is a soliton envelope, and the dotted line, red dotted line, is this triplet bound state. So when we look at the redox processes, we know that this is a sequence of polypeptides, enzymes, localized within the bilipid layer of mitochondria, for instance. And there are some enzymes with very large mass. They are shown on this picture on the top of the slide in green. In blue, these are small enzymes which are easily movable in the within the cell. But the green enzymes are localized. And there is the sequence of these proteins so that the electrons are transported from the first to the second to the third and to the fourth polypeptide. First, second, and third polypeptides are, the next slide, please. They do not have a local magnetic field. So within these enzymes, electrons are transported by Soliton mechanism. Their spins are anti-parallel. While when they reach the fourth, the last enzyme, this is cytochrome C oxidase. It contains the groups in which ions of ferrum are localized in such a way that their magnetic fields, their magnetic moments are parallel. So there is the local magnetic field. And according to our estimate, this field is enough to reorient spins of electrons, so that in this cytochrome C oxidase, we expect that electrons will be propagated by in the triplet bound state. And this has been in fact confirmed by Hall effect. So we think that that was one of the experimental evidences of the validity of this theory. The next slide, please. I did not tell you about the Coulomb repulsion. Well, we can take into account the Coulomb repulsion. Soliton is localized within several unit cells, within several peptide groups. Its charge, electron charge, is screened within our system. So the Coulomb interaction can be taken into account within the perturbative method. And this account shows that if there is no Coulomb repulsion, or if it is very weak, then our potential well has one minimum, and respectively, the envelope of the soliton had one maximum. With the increase of the Coulomb repulsion, the one minimum potential well can split and become a double well potential, with some distance between the two minima, and this distance is less than the width of the potential well. As a result, the soliton can have, the soliton envelope can have two maxima. And what is important is that bisoliton is stable only when its bound energy is big, binding energy is bigger than the energy of the Coulomb repulsion, which in terms of our parameters is possible when the nonlinearity parameter, when the electron lattice coupling is bigger than some critical value. And we know the expression for this critical value. The next slide, please. This result, we suddenly found the comparison with the numerical experiments, numerical results done by other people, Четвериков, Velarde. They had a numerical simulation in the discrete lattice of the system when they inject 2 electrons in the lattice. So the envelope of the probability of localization of electrons is shown by green curves. Let us look in the first row from the left to the right. And then we go to the second row. So we see the green, we follow only the green light. In the beginning, they had the nicely localized soliton-like state. Then they increase the Coulomb repulsion. And they see that there appears some internal structure. The maximum has some oscillations. The third figure, even stronger Coulomb repulsion. Now the maximum splits in 2 maxima, and this maxima, distance between the 2 maxima increases at even stronger U4 Coulomb repulsion. In these panels on the right, in each panel, I show the results of our analytical results, changing the strength of the Coulomb repulsion. And the ratio between U1, U2, U3, U4 is exactly the same as it was in the numerical simulations by Четвериков and Velarde. And we saw that these blue curves reconstruct very nicely the numerical results. Of course, this is continuum, analytical model. Analytical model is continuum, so we do not see oscillations, small oscillations, which appear when we take into account the discreteness of the system. The next slide, please. Now, solitons exist not only in proteins. There is a large class of quasi-one-dimensional systems, some of which I show on this slide. These are organic quasi-one-dimensional compounds like this BDT, BTS salts. There is the system called polydiacetylene, with the periodical structure formed by 4 carbon atoms in a unit cell. There are conducting platinum chains, salts of transition metals, conjugated donor polymer acceptor systems. In these systems, the solitons are formed in some cases at moderate doping, like in salts of transition metals or in superconducting BDT, TTF salts. Next slide, please. In this case of the conducting polymers or conducting platinum chains, the number of electrons is, of course, not 1, not 2, but is relatively big. So we have to take into account the interaction of a bigger number of electrons in our systems. And we have shown this. There is a misprint, cnoidal, it should be cnoidal wave. At a large number of electrons in the systems, they form the cnoidal wave, described by elliptic Jacobi function, dn or cn function. And the energy of this cnoidal wave depends on the density of electrons in the system. It is a non-monotonous function. It turns out that there exist critical and optimal concentrations of electrons in the system. When there are, I told you that it is energetically advantageous for electrons to create the common potential well. this increases their binding energy. So the energy of the system first increases, but then the wave functions of separate, isolated, we can say, bisolitons start to overlap. And this overlapping leads to repulsion between them. As a result, the energy after certain concentration starts decreasing. And finally, there is some concentration when electrons cannot form the many soliton cnoidal wave. Well, the cnoidal wave exists, but it looks already like the plane wave, which means that electrons are in almost free electron state. This theory we have applied to explain the dependence of the critical temperature of superconductors, for instance, of the high temperature superconductors, on the concentration of charge carriers, on doping. And in the lower figure, we can see critical temperature as function of concentration. Lines correspond to our theoretical model. Points, symbols correspond to experimental data. The upper curve is for yttrium systems and the lower is for lanthanum compounds. The next slide, please. Leonas, could you please stop me 5 minutes before the end of my talk?
Yes, I will do.
Thank you. Now, another question is, so, solitons, do they always exist in low-dimensional systems? A good question. It would be strange if they always existed. To answer this question, we have developed the variational approach. From the general theory, we are aware, we know that in the electron phonon system, there can be different types of ground electron state. It can be small polaron, it can be almost free electron, and it can be this soliton about which I told you. And let me explain you that the soliton, Davydov soliton, in linear approximation corresponds to a large polaron. So we have shown that the question which type of the ground electron state is realized in the system depends on the strength of the characteristic parameters of the system. When we have acoustical phonons in the system, we have this non-linearity coupling constant G, and we have non-adiabaticity parameter, which is the ratio of the electron resonant interaction to the velocity of the sound waves. Similar parameters we have for optical phonons. The next slide, please. And we have built the phase diagrams which show which type of the ground state is realized in the system, depending on these two parameters, G and gamma. Here, SP corresponds to the values of the parameters when the small polaron is the lowest energy state, AFE is almost free electron, and Sol is soliton. The left figure is for the case of acoustical phonons. The central figure corresponds to optical phonons, and the last one is acoustical plus optical phonons, of course, at some fixed values of one of the branches. With these diagrams, we have analyzed that systems which I showed you, low-dimensional systems which I showed you before. And we predicted in which systems the electron transport or conducting properties were determined by solitons. Sorry, we have by almost free electrons or by small polarons. The next slide, please. Now, the important question is the structure and geometry of the system. When we take into account the more complex structure than on that figure I showed you on the second slide, like for instance, alpha helix has 3 polypeptide chains and it has helical structure. So in these systems, there can be different types of solitons realized. For the alpha helix, we have shown that there is no energy gap in the system. The lower band is almost degenerate. It has minima, equal minima, but displaced with respect to the wave number. The next slide, please. And from which of these energy band the soliton level can be, the soliton can be formed. The blue line on the left figure shows the isolated soliton in a single molecular chain, polypeptide chain. The red line corresponds to the energy of the soliton split from the upper energy band, which I shown you before. The yellow line from the lower energy band. And according to Young-Teller theorem, there can be formed the hybrid entangled soliton state, and its energy is much lower than the energy of all these other solitons. In fact, it is for the parameters of the system, it is 50 times lower than the energy of the isolated soliton. And respectively, the envelope of the soliton has a more complex structure. The black curves show the probability distribution on each of the 3 strains, the polypeptide strains. The red curve corresponds to the sum of these 3. The next slide, please. This soliton has internal structure, it oscillates. Lifetime of the hybrid soliton is much higher than the lifetime of isolated soliton. It is 3 orders of magnitude higher. And in fact, this explains the experimental data on the lifetime of amide-1 vibration in 3 very close systems, in myoglobin, in yellow polymer, and in L-alanine. The primary structure is the same. This is L-alanine unit. Myoglobin is essentially alpha helical system. So the lifetime in it is 400, 500 picosecond, which is much higher than the lifetime in the yellow polymer, which is beta sheet, and this is still bigger than in isolated. The next slide, please. Another important phenomena is that the discreteness of molecular chains is very important. Due to the discreteness, there exists the periodic Pierce-Nabarro relief in the system. And as a result, the consequence of this Pierce-Nabarro relief is the periodic time dependence of soliton velocity. This soliton velocity is determined by elliptic Jacobi function dn, which can be split, which can be expanded into series of harmonics with the frequency of the main harmonic omega, which is proportional to the average velocity of the soliton per period. This has very important consequences for biological systems, that depending on the physiological processes, the frequency of the oscillations of soliton depends on the velocity of electrons, and electrons radiate electromagnetic field. So the frequency of this electromagnetic field depends on, is physiologically important. The next slide, please. This shows the experimental, the numerical computer modeling. We see there are the small oscillations of the amplitude. On the right-hand side, we see the oscillation of the center of mass coordinate caused by this phenomena. And another important thing is that the height of this barrier, of potential Nabarov barrier, depends on the soliton amplitude. It depends on the, again, on the nonlinearity parameter G. Two narrow solitons can be pinned by the potential, by the Peirce-Nabarov potential. The next slide, please. I am afraid I have to finish soon. The result of this Peirce periodic Nabarov potential is that solitons can, that the ratchet phenomena can take place for solitons. Ratchet effect is the appearance of drift under the action of external periodic force. For instance, we know that the ratchet devices are used in cars to change the rotational movement of wheels, for instance, in the car, to the unidirectional movement of the car. Under the action of the external electromagnetic field on molecular lattices, soliton also can attain the drift. The next slide, please. On this slide, we see the oscillations of the soliton center of mass coordinate. Not only oscillation because of the dependence of the velocity, but also the drift. We see that the central mass, the coordinate of the central of mass, moves from the site number 100 to some 120 and so on. And this propagation can be in one or another direction. The next slide, please. This phenomena is critical with respect to the frequency and intensity of the field. And what is also very important is that the stochastic resonance takes place for solitons. Depending on the temperature, the drift, the amplitude of the drift can first increase and then decreases with temperature. And this dependence has the lambda type curve, as shown on this figure. This is the drift, the figure shows drift, amplitude of the drift of the soliton as function of temperature for different values of period of the field or for critical or amplitude of the field. The next slide, please. I told you about pinning of solitons. So there is the critical intensity of the field below which the solitons, narrow solitons, are pinned by the lattice. But at higher, they start moving, the drift increases.
5 minutes.
Thank you. So the critical values of intensity of the field and period of the field decrease with temperature, which is as shown, for instance, in this by this number, critical intensity of the field at zero temperature is 0.9. At temperature in some adimensional unit 0.1, it is 0.6 and then 0.3. The next slide, please. Very briefly, the Soliton mechanism we have applied to explain the delayed luminescence from biological systems. I will skip the scheme. The next slide, please. We managed to explain not only qualitatively but quantitatively the kinetics of delayed luminescence from unicellular algae, acetabulary acetabulum, shown on the upper part of the figure. We have predicted the nonlinear dependence of the quantum yield of delayed luminescence on the intensity of the illuminating light, and this has been confirmed experimentally. I will skip the next slide, please. We have tried also and managed to explain some data on the existence of solitons or small polarons in zigzag nanotubes. The next slide, I will skip this, skip another slide, please, another. Other experimental data I already told you during the talk. Thank you for attention. One slide more. Merry Christmas and Happy New Year. If I have still some minutes, I will be glad to answer the questions.
Okay, thank you. Questions, please. Okay, so maybe I will ask you. So
yes,
biological systems, they are flexible. So practically it could be understood in terms of the disorder in various aspects. So what is the possibility? How does it affect on the solitons, the disorder?
Yes, the disorder, well, there are different aspects of the disorder. For instance, in the disorder can be because of the temperature, the oscillation of units with temperature. We have taken this into account and have shown that if the temperature is lower than the critical value, then a certain value, the Soliton mechanism improves, is more effective, and with further increase of the temperature, increase of the disorder, the effectiveness decreases, while at critical temperature solitons break down, and deformation and electron become separate and non-interacting. Another level of stochasticity can be, well, it's not really stochasticity, but different masses of amino acids in the system. And there was a large investigation by Boris Malamed of these aspects. Another stochasticity, this thermal Y noise, which I told you already when I told about the ratchet phenomena. There can be also stochasticity caused by external electromagnetic field. This needs a serious investigation. The same as the role of the temperature, because here it is very important not to forget the physical system and not look into mathematics only.
Yeah. Okay, so thank you. Anybody else would like to?
I see that Volodymyr Kravchuk has the question.
Yeah, I have a question about stochastic resonance for solitons.
Yes.
So typically stochastic resonance appear in when you have a system with double well potential and escape time from one well is become comparable with period of driving. But about soliton, how you get this double well or many well potential for a soliton?
Yeah, not necessarily double well potential for the soliton. For the soliton itself, the double well potential is formed when this is the Pars-Nabarro potential, when we have, for instance, two atoms in a unit cell. If these atoms have different masses, then we will have a double well potential, Pars-Nabarro potential. If we have the one atom or a symmetrical chain, molecular chain, then the ratchet and stochastic resonance can appear when we apply the biharmonic or more complex electromagnetic field of more complex structure, periodical with zero mean value, but with several harmonics, so that the necessary conditions can be in principle satisfied.
Okay, so thank you, and probably we have to stop here because we are.
Thank you very much.
Out of time. Yeah.
Bye.
Thank you. Thank you. Can you switch off your presentation?
Sergey, could you please switch off my presentation?